Hi, I'm Sean O'Brady from Corporate Communications, here for another episode of NRTV. Today we're in the Niagara Region Environmental Laboratory, here to talk to Norma Linkowitz about what they do here. So Norma, could you please tell us a little bit of what you do here and what kind of water that you're treating, or what type of water that you're looking into?
Hi, Sean. We are the Niagara Region Environmental Laboratory, and here at our facility, we strictly test wastewater samples that come from the wastewater treatment plants, and we also test biosolids from the biosolids storage facility that Niagara Region has.
And what type of water comes in? Do you get the water that comes in just after it's been treated, or the water before it comes into the plants as well?
Actually, we get both samples. We get the samples that are coming in before treatment, and we get the samples that are coming in after treatment. And what we're trying to do here is make sure that the treatment process is working properly.
I think we've got a couple of samples here. What sort of samples of wastewater do we have here on the table?
This is from one of our wastewater treatment plants in the Niagara area. This is what is coming into the wastewater treatment plant, and this is what is going out into the wastewater treatment plant. So here is a good indication of how the cleaning process is working. So this is what is regulated, what is going back out into our lakes and rivers.
And do you test both of these liquids?
We do test both what is coming in and what is going out, but what is most important is what is going out. We don't really have control of what is coming into the wastewater treatment plant, but we definitely have control of what is going out.
All right, so now we're at another station here. Maybe you can tell us a little bit of what we're looking at here. I believe this is the area where we're going to be starting to test some of our wastewater.
Yes, over at this station we test our wastewater for total suspended solids. Total suspended solids is exactly what it says. We're collecting the amount of suspended solids on a filter paper, and this is also a test that is regulated by the Ministry of Environment. All of the wastewater treatment plants have to fall within a specific limit, and Colleen here, our analyst, is busy analyzing and filtering all the wastewater samples that come into this place.
Okay, well now we're at another station here. Another station also looking at some of our wastewater that we're looking to test. What exactly is going on here?
This is a segmented flow analyzer instrument, and today we're measuring total phosphorus and total Kaldol nitrogen on this instrument. It is a colorimetric reaction that takes place at this instrument. So the sample is introduced, a sample needle mixed at various integrated times with different reagents. A color solution is formed and read on a photographic spectrum.
And why is it important to test for this material?
It's very important that we don't have huge amounts of phosphorus and nitrogen going back into our environment. Again, that's why we're always trying to make soaps that are low in phosphorus and nitrogen, because it ultimately affects our aquatic life, and we're trying to make the water as safe as possible. So we don't want all these contaminants back that are discharged back into our lakes and rivers. So right here we have the setup for E. coli. The samples are received from the wastewater treatment plant in this bottle. This bottle is then poured into this bottle with this little capsule. This capsule contains reagents, and the sample is mixed. Then it is poured into this tray. After this tray is filled with this bottle, it is sealed. It is placed in this sealer, and it is incubated for 22 hours. After the 22 hours, we take it out of the incubator, and we are observing the sample for total coliforms. As you can see, we have a color change. After the 22 hours, if there is color change, we see these yellow squares appear, and this indicates total coliforms are present. So we know that total coliforms are present, but we don't know if E. coli is present. But
How do you figure out if there's E. coli present?
if there is E. coli present, the samples will fluoresce, and the fluorescence is read in this little box here. And if you take a look, you'll see these little squares that fluoresce, and again, the lab technician will count the amount of squares that fluoresce and do a calculation, and based on the calculation, we'll see whether the wastewater treatment plant is in compliance.
So this is interesting. So I can see that some of these things have fluoresced, so I can see that they're changing, so I guess that means there's going to be E. coli in there. What happens here at the lab if you're performing one of these tests, you do find E. coli, you do find some other materials? How do you relay that information back to the plants, and what happens from there?
What we will do if there is an exceedance, we will notify them immediately, so that way they can do any operational adjustments that are required on the spot.
Well, wonderful. Well, thank you so much, and thank you for providing that great tour of the lab here. Wonderful. This has been Sean O'Brady here with NRTV at the Niagara Region Laboratory with Norma, keeping our wastewater clean and our environment safe. Thank you very much.